Welcome to BioGenerator. I'm Jim McCarter, Senior Managing Director. BioGenerator Ventures creates, builds, and invests in companies that are solving important challenges in human health and agriculture. We are the startup arm of BioSTL, a nonprofit that for 25 years has laid the foundation for the growing innovation economy in St. Louis. BioGenerator has now invested over $50 million to help start 140 St. Louis biotech companies. Our companies have gone on to raise over $2.9 billion in follow-on funding and created thousands of jobs. We've also supported over 200 companies with office and lab space here in the BioGenerator labs in Cortex and at the Helix Center in 39 North. So we're delighted that you're here with us today for our 10th Innovators Hour. We hope that you'll consider joining us again in the coming months. In November, we'll host Erica and Andrew Barnell from Genoscopy, and in December, Andrew Young and Allison Mayer from WashU, BioGenerator, and Paradex. Both of those companies are in cancer diagnostics. And we've begun to fill out the calendar for 2026. So in January, we'll be joined by Stephen Von Rump from Sweet Spot Digital Health, and in February, John Silva from WashU and Centiar. So search Innovators Hour at BioGenerator for the website and more information, including videos of prior conversations. So for today's Innovators Hour, I'm pleased to welcome Cory Berkland, PhD, Professor of Chemistry and Biomedical Engineering at WashU and co-founder of multiple BioGenerator portfolio companies, including Bond Biosciences, Kinemune, and Axioforce. And previously, Cory was co-founder of Orbis Biosciences and Savara Incorporated. So Cory, you have an unusual background geographically and educationally for an endowed professor at WashU. And so perhaps you could tell us a bit about growing up on a farm in Iowa and what you didn't know about careers in science growing up, but what you did know about running a small family business.
Yeah, that's a great start. So I'm Cory, and I'm from Iowa. How's that for a good start? I grew up in, if you look on a map, and especially a map at night, and you look where all the lights are, you see cities. And if you look at the darkest part of the map in Iowa, that's where I'm from. So it's the northwest part of the state. The closest town was 10 miles from our farm, and it had 80 people. And that's where I went and played baseball. We had 9 players. If somebody was sick or didn't show up, we drove to their house and picked them up and made them stand in right field. And so I didn't have much exposure to education. My school was small. I think I told you, Jim, on a preview to this, the highest level class we had was pre-calculus. And so I didn't necessarily feel academically prepared as I was going through school, but I did feel like I had, enough. I had enough exposure to physics and chemistry and math that I was prepared. What I didn't know at the time was that I was developing some skills for running a small business. When you live 10 miles from the nearest town, you end up having to be really resourceful. So if something breaks, you have to fix it. You can't go down the road and find somebody to fix it. If you go to town to get a part, you might waste over an hour going to town, get the part, you go back, it's the wrong part. And so you're stuck, and you go back again, and you learn to be very efficient with your time and the way you choose to spend your time. And I think I wasn't appreciating it in the moment, but I was learning how to use resources well, how to be very efficient with my time, how to manage money through a very difficult period of time with farming in the 90s.
Yeah, and so as you got started on your education after high school, you were 2 years at junior college and then transferred to Iowa State. And it was some point in that process where you realized that you had a knack for science, or someone told you that this was a possibility for you. What was that like?
Yeah, my life is not really including a lot of forethought up to this point. And I knew I wanted to play baseball. So I went to the local junior college and I played baseball for 2 years. And along the way, as you mentioned, somebody said, oh, you should tutor physics. You did well in physics, so you should tutor physics. And I needed money, so I was tutoring physics on the side, mostly to other athletes because they weren't doing well in the physics class. And I also was doing well in chemistry and math, and somebody said, well, chemistry, physics, math, you should be a chemical engineer. I was like, oh, okay. So after 2 years at junior college, I applied to Iowa State. But back then, we didn't really look around for colleges and what was our best fit. You just went to the state school. And so I knew it was a good engineering college, and I went to Iowa State University. I'm sorry this isn't more compelling in the really thoughtful approach to living life.
And then when you were at Iowa State, did you have any experience in the lab, or did that come at Illinois during your PhD work?
Yeah, when I got to Iowa State, I found out that I qualified for a work study program because of coming from the farm and not having much economically. And I was asked if I wanted to join a laboratory. And so I said, sure. It'll replace my physics tutoring income that I had at the junior college. And so I started working in a laboratory. And again, this doesn't really sound like I had a lot of thought going into my life at this point, but I met graduate students and they said, hey, you're pretty handy in the laboratory. And I was like, yeah, that's because I worked on tractors and repaired fences and took care of animals. So I knew what I was doing in the laboratory. They said, hey, you should really think about graduate school. I was like, oh, I've never heard of graduate school. So I applied to graduate schools in the Midwest. And because of my research experience and a couple of posters and things that I have a publication record, I was able to get into the University of Illinois for chemical engineering graduate school.
Excellent. And so one of the things you mentioned, you were thinking of okay, this may lead to a career in teaching, but there was a visitor to your lab in Illinois that I think your advisor, your PhD advisor, had a collaboration with industry. And this visitor made a comment that seemed to trigger something in you about this idea of having impact. And if you want to tell that story, I think it's a pretty interesting story.
Yeah. So I was not as good academically as some of the other students. Illinois we had the #1 math student for his age in the entire country of India. And then we had very talented students from China and Indonesia. So my buddy from Tennessee and I would go study with them and try to do well enough on the test to pass. So we managed to make the grade academically. But the interesting thing was when we went to the laboratory, it was almost flipped. People were coming to my buddy from Tennessee and I because we knew how to take equipment apart and understand how it worked and repair things in the laboratory. And so along the way, I got good at doing a particular type of research. I was microencapsulating drugs. So I had built these little devices to make beads, very uniformly sized beads, and I could control the bead size with just a push of a button because I had rigged up this vibration system and electrocuted myself several times. But I could titrate the bead size, and then I could use that to control the rate that a drug would come out of the bead. And I didn't know this at the time, but my advisor was selling this to pharmaceutical companies. And so he told me, I have some good news. We have 2 industry sponsors for your research. And I actually felt kind of dirty. I don't know if you remember being an undergrad. I barely do. But you kind of develop almost this attitude of well, I'm going to publish things and I'm going to write a book chapter. And that was my contribution to society. And so when somebody came in and told me that Big Pharma was paying for my PhD, I was almost offended or felt it I needed to change my jacket. And one of them came up to me and said, what you're presenting to us is going really well. And we're really excited about the potential to make a product for our company. What are your plans for your last year of graduate school? And I said, well, I'm going to publish a book chapter, and I'm going to write a paper, and I'm going to defend, and that's probably about it. And this guy noticeably became agitated by my answer. He put his hands on his head, and he said, you have to patent your work, and you have to maybe start a small business or find some way to continue partnering with industry. And I swung from this really altruistic attitude about research. I didn't become a patent jockey, but I swung all the way over to the other side, like, oh, this is actually how I would like to have impact. I would like to help make medications that get to people and impact human health. And maybe I'll sort of deprioritize the impact factor of the journal I'm trying to publish in.
And I'm going to skip ahead for a second because we'll get to your work at University of Kansas and all the companies that you founded. But I think one of the interesting things is that you're a fairly recent addition to the WashU faculty. And in your endowed professor talk, this was kind of the point of your talk, right? Was that rather than just doing science and publishing it, that you're trying to leverage that to have impact in the world. And you're using commercialization and innovation as a vehicle toward that impact. But how is that resonating with your colleagues at WashU?
They've been very accepting. They actually knew that about me coming in. It's part of the reason I was recruited. I was nervous when I got my first job because I was thinking it was going to be all about scholarship and that measurement of impact. And it turned out it is. You have to check that box and you have to do that well. But if you do that well, you can also have broader impact. Impact with startups, jobs, creating maybe even products.
Right. And so let's talk a little bit about, you joined the faculty at University of Kansas around 2004. You started a company in 2007 called Orbis, and that licensed the microencapsulation technology that you'd helped to develop at University of Illinois. And so as we were talking about Orbis, it was sort of the opposite of an overnight success. I mean, and so if you can tell us sort of the story of, how do you keep a company going and, until it has the chance to really blossom, and what are kind of the fights along the way to make that happen?
It's funny, you read books about companies and that's when you discover that none of them were, were an overnight success. It's the company that, was an overnight success after 15 years of work on the company. And that's the story of Orbis. So I actually started that company with my advisors at Illinois. It was called Airy Sphere, which airy in Korean means happy sphere. And we had a little bit of seed capital from a small boutique venture fund, and it went nowhere. And the whole thing just kind of fell apart. I didn't know what I was doing. And then I left and got a job at the University of Kansas, so there was no longer a champion. And once I got to Kansas and I found I could publish papers and get some grant money, I was like, oh, I should really go back and turn that into a business. So I found a gentleman at the Kauffman Foundation. We were both in our late 20s and thought we knew everything. So we founded a company, Orbis Biosciences, and we were able to get these small awards from industry. There's a solicitation called HALO right now, which puts out industry challenges. In our day, it was called 9 Sigma. There was one called Innocentive. And it's just challenges from big industry about how do you solve problems. And we started applying and winning. And we're like, oh, people must really want this. They want microencapsulation. So we started building the business, and yeah, it was a grind. Like you don't know what's going to fail until you actually start doing it. And then we saw failures in scaling up and failures in satisfying customers with the right product profile. But persistence sort of won the day, and we ultimately sold that company in 2020.
Yeah, and so one of the things I think you determined was this trying to figure out what the customer, in this case, the pharmaceutical companies, figure out what they need and what they want. And you figured out that it was not just research and licenses, but it was also some manufacturing capability. What was that kind of mix that made that company attractive?
Yeah, it's a great question. We were really good at going to conferences and shows and getting introduction into scientists at Merck or Pfizer, and then going in and reminding them how much they loved us and really needed microencapsulation. And we were getting checks for $50,000, $100,000, sometimes $250,000. And it would be like a 1-year R&D project, and you had to deliver on certain proofs that you could microencapsulate and control the release rate of their medications. But then when it came to convert into a development project, we only had plans. We didn't have equipment that could produce a phase 1 GMP supply of material. And we kind of were almost developing this reputation of, after doing that 10 times, 20 times, that we were a pier instead of a bridge. And fortunately, we submitted a large grant application to the NIH called Lab to Marketplace, and they helped pay for the engineering and design of a phase 1 GMP microencapsulation facility that we built in Lenexa, Kansas. Once we showed people that we could get them into a phase 1 clinical trial, that still wasn't good enough. We were like, but we did it. And they said, well, you're gonna have to show us that you can conduct a clinical trial. So then we got another grant and we conducted a clinical trial. We were able to taste mask a steroid. Not jaw-dropping science, but quantitative and very difficult to do. And we got the feedback from that. We took that back to customers and they said, well, that's great, but now you're gonna have to show us that you can get a license with it. So every time we tried to sell the business, there was one more thing to do. Yeah, we ended up getting a license with the makers of Mucinex, and we made a long-acting oral suspension of cough and cold medication for them. And we got a second license with a women's health company. And once we had all those pieces pulled together, we were able to sell the business.
And so another one of the companies that you started along the way then was Savara. And what's the story behind that one?
So we started Orbis at the end of 2007, and it took us until 2020 to sell that business. But evidently I'm a glutton for punishment, because in 2008, I started another company. We were doing particle engineering that wasn't microencapsulation. It was a nanotechnology, which at the time was the flavor of the day. And we showed that you could take existing dry powder inhalers and you could improve their efficiency by like 4 and 5 times by using a sort of a nanostructured particle that flew really well when you inhaled it versus gritty sort of micronized particle that just ends up crusted in the device and never goes into your lungs. And we had a new faculty member who I was friends with, and her husband was an MBA student and needed a project. And so we started Savara Pharmaceuticals together as part of his MBA. That was in 2008. And then in 2010, we had our first financing, and fast forward to 2017, and we went public on the NASDAQ.
Excellent. And so
there was some trouble along the way.
Yeah, I imagine there's do you want to fill that in more before we jump into more recent
I don't
history?
know, I might get PTSD if I talk about that one too long.
Well,
so,
yeah, go ahead.
Yeah, I'll just fill in some of the blanks.
Yeah.
So it was a partnering model again, because I was really good at that. I was good at going and talking to pharma partners and coaxing them into a deal. And so we had 3 large deals to reformulate existing aerosol products, like think about Symbicort and Advair and Pulmicort. And we were showing that you could cut the amount of steroid a child would have to inhale by 80%, but they would still get the same dose into their lungs. They just weren't getting this other exposure through swallowing and stuck in the mouth and everything. And then 2008 and 2009 came, the financial crisis. And everybody sold those businesses. All of our partners got acquired or divested, and we were done. I mean, we were toast, because that was our income, plus some grant money and economic incentive money. So we had to pivot and create our own product, which was an inhaled antibiotic for treating MRSA lung infections, drug-resistant lung infections. And then we ran that phase 3 clinical trial. And in the meantime, we had partnered with another business. We had actually acquired a second business that had an inhaled growth factor, or GM-CSF it's called. It's a cytokine. And we were putting that in second place because we had this lead. And then we showed definitively in a phase 3 clinical trial that we could cut MRSA in the lungs of patients, but the patients showed no clinical benefit. They weren't able to breathe fast, which about made me lose my marbles. And so the FDA said, well, you can rerun the trial and try another course of antibiotic and see if people can breathe fast after 2 courses. Well, that's like another $150 million in 3 years. So that was dead in the water. But luckily, this other company we had acquired was successful. So that was, the 2 assets together were enough to take us public. And yeah, don't look up the stock ticker because it goes like this, and then it goes like this, and then it goes like this, and like that. And finally, we're like right about here. And I can sell some shares maybe at some point.
Well, so let's turn to more kind of recent history. And so I want to talk with you about the 3 companies that are part of the BioGenerator portfolio: Bond, Kinemune, and Axioforce. Well, let's start with Bond. So the way that you described Bond to me, or sort of the start of it, was unusual. It sort of began as sort of a thought experiment around how to create, a successful company. But rather than starting with, oftentimes people, especially academics, will start with the technology and then search for an application. Or sometimes people will do maybe one step better. They'll start with an unmet clinical need and then try to find something that meets that need. But Bond started with a somewhat different approach.
Yeah, I was tired of getting beat up by pharmaceutical partners because through all those partnerships, people wouldn't pay on time. They'd complain about the results. They'd divest their business and just destroy my business. And I was getting sick of that. So I decided to try a foray into a real drug company instead of a partnering drug delivery company model. But I knew enough about investment that I knew I should try to answer all the questions first. So you've all probably heard of due diligence and the due diligence memo, and you have to answer all the questions that investors are going to ask. I had this idea of trying to reverse engineer the due diligence memo and just have the highest probability of success in every category. And so that's what I did. That's why I started with sort of that thesis and tried to reverse engineer Bond.
Yeah, and so then I think you started with, why do products fail, right? And so safety and efficacy. And so what did you do to kind of think that through?
Yeah, so half of drugs fail because of safety tox. And I started looking up, well, what are the companies that have the least risk from a safety tox perspective? And I convinced myself it was drugs that you take that never go into the rest of your body. They just are oral drugs, and then they go, right out the back door. And the worst thing that could happen is you get constipation or something. So that I thought, okay, if I make a non-absorbed drug, then when people evaluate this for investment, they'll be like, oh yeah, that looks pretty good. And then efficacy is generally the other half of failures. And so we had to try to match this profile of where the drug is with what it does to show a clinical effect. And we sifted through other drugs that are non-absorbed. So there's approved non-absorbed drugs that treat hyperphosphatemia. If you have end-stage renal disease, you get too much phosphate load in your body. Hyperkalemia, too much potassium, same situation. There was drugs approved to lower your cholesterol, and they work by actually just binding the bile acids in your gut, which is a precursor to cholesterol, and you can bring down your cholesterol levels safely. And in reading the patent literature, I saw that people were also trying metals. And I was like, oh, well, I've worked on metals, and I was drawn to this concept of binding to iron for people who have iron overload disorders. They hyperaccumulate iron because of a genetic defect. And if they would just take this concept of a pill that binds all the iron from the food and keeps it in the GI tract and then expels it, maybe I could treat those patients. And so that led to a clinical evaluation. So people want to see what's called hard clinical endpoints. Measuring iron is a very hard clinical endpoint. So I felt like I had some momentum in terms of a thought.
Yeah, and in terms of the patient benefit, what's the current therapy, and how would the therapy developed by Bond be better?
The standard of care right now for patients with iron overload disease is either lifelong bloodletting. So you basically go in and you have what's called the therapeutic phlebotomy intensely at the beginning when you're first diagnosed with the disease. And then once you're well managed, once you have a normal iron level in your body, it's roughly quarterly for the rest of your life. You go and give a unit or two of blood, you have to go to a specialist. Or there's another form of the disease where you can't give blood because you're anemic or you have some other condition. And then you have to take nasty systemic iron chelators. And you've probably heard of like iron chelation and demetalling people, and that's horrible. And those drugs all have black box warnings for side effects. And so we felt like we had kind of carved out this really interesting niche of an opportunity.
Yeah, and so where is Bond today?
So Bond is raising a Series C. We have a lead investor on a term sheet for a Series C financing, and that will progress us into a multiple ascending dose safety study and a first in-patient efficacy study. We've completed and published a single ascending dose clinical trial.
And you've gotten this far with a pretty small team. What's the kind of footprint of that, or is that not true?
That is absolutely true. So Danny Griffin, who is a venture fellow here as well at BioGenerator, helps me with business development and some of the operations. Has been involved in our financing discussions as well. Me, who may or may not get paid depending on how far we are from our previous financing, and I consult for the business and generally lead what's called CMC, so the manufacturing, the drug, any biological studies, non-clinical work in animals and so forth. And then we have my co-founder, Mike Riley, who I think is currently the chief executive officer. We just tag in and out with some CEO titles or COO titles. And he runs all the finance, daily operations, contracts, intellectual property.
Yeah, but you've gotten a long way with a part-time team and sort of a virtual company. Yeah,
It's a pretty good model, though. A lot of venture studios or smaller funds will try to do this like chess match of bringing in people at the right time, because you'll go through a really intensive phase where you have to manufacture and qualify the drug. Well, you don't need your chief medical officer, and you don't need your non-clinical biology head. Sorry, Danny, but you might not need business development real hard during that time. And so you're always like trying to juggle in the right people at the right time and cost conserve. That's like driving to town to get a part for your tractor.
Right.
You're just trying to like,
but not having sort of a fixed payroll where you're going to have to pay this 10 or 20 person team kind of on a perpetual basis, where they don't have enough to do, right?
Yeah, burn is the killer. Yeah. Financial burn, it can be troublesome.
Yeah, so do you want to talk about Kinemune next or Axioforce?
Sure, let's talk about Kinemune. Okay. Yeah. So Kinemune, I had a friend who passed away from cancer. She was one of my wife's friends from high school. They only graduated with 33 people in their class. She argues with me and tells me it's 35, and I'm like, it doesn't matter if it's that you graduated 33 or 35. And she became an oncologist. Tall, beautiful young lady, played basketball at a couple of small schools, and then went to KU Med and became an oncologist. And as ironic as this seems, she ended up in Lawrence, Kansas, the same town we were in, coming from this small rural Iowa town. And she was the head oncologist as a new doc. And about 3 years in, she got diagnosed with leiomyosarcoma, a really rare, slow-growing sarcoma that they found first on her spinal cord and then disseminated in the rest of her body. And I don't fear stepping into new research areas, because I feel like, as Doug would tell you, as a chemist, you're good at chemistry. And you maybe don't think about not being an oncologist or not being a neurologist. You think about the problem to be solved from a chemical or engineering point of view. So I was like, you know what? I'm going to do oncology. And at that moment, when she was in her last couple of years, we decided to figure out as pragmatic a way as possible to get something into the clinic that you could inject in and around tumors and give people a chance who don't have any other options. To watch her go through the last year of her life, and she's gone through all the therapies, and now she's just stuck, is a difficult thing to watch.
And so what was the insight behind the company in terms of what are you going to inject into that tumor that's going to do something different?
Yeah, Danny again, and Grant and some of the people and Bryce that were in the lab, that all these things that were going on, sort of centered around, at the time, autoimmunity. But we really followed immune responses closely in what's called the experimental autoimmune encephalomyelitis model. It's the multiple sclerosis mouse model. And we couldn't figure out why this approved drug called Copaxone worked, because you inject this drug and there's none in the blood. So it's an MS drug that reduces relapses or events in MS, but yet it's just sits at the injection site. And so we became kind of intrigued by this drug's mechanism of action, and we injected some in a mouse, and we pulled back the injection site, and we found that this mixture of peptides that's supposed to look like the coating on your nerves just crashed out. It just precipitated at the injection site. And about 75% of the time, there's a lot of swelling and irritation at the injection site. And so we kind of have this light bulb moment. Well, if it causes inflammation where you inject it, maybe we should inject it in tumors. And so we started doing that in mice, and we started seeing, like, yeah, it causes inflammation. We don't know if that's enough, but it's interesting enough that we studied it in tumors. And then we wrote a letter to the FDA and asked if we could take that drug and inject it in the tumor of patients, real patients. And we conducted a clinical trial with Kansas Medical Center, where patients who came in and had a melanoma or a head and neck cancer, and they were scheduled for removal, resection of that cancer. In that 30-day window, we could inject in and around their tumors, and then we could compare the biopsy they came in with to the resected tumor and see if we're doing something, if we're activating an immune response. And it was kind of exciting. We were seeing immune cells, like, suddenly that were avoiding the tumor were starting to creep into the edges of the tumor and look around a little bit. So yeah, an exciting moment for the lab, and it led to the founding of Kinemune.
And so what's next for Kinemune?
So we actually tried to just repurpose that drug and get partners interested in working with us to further that evaluation. But generally people aren't interested in a drug that's going generic, now being repurposed for injection into tumors. So we've multiplied our approach by pairing that, first whittling that drug down into a new chemical entity that still kind of sticks in the tumor and immunostimulates, but now matching it with another ingredient that really turns on the immune system. So we would call that like an immunostimulant that's trying to drive T-cell responses and really turn on recognition of the tumor.
Great. So let's turn to Axioforce. Very different story, and maybe the least biological of the things we've talked about, but actually some of the applications are biological. So
Jim, I have this history. I was in my second year at Kansas, and I was a therapeutics guy. I'm going to work on pharmaceuticals. And then somebody said, well, we can get a grant for biomaterials. I was like, I don't like that idea. And they said, well, we have money. And I'm like, oh, okay, that's a good idea. And then the next year, my third year as a professor, it was chemical and petroleum engineering, which is one of the departments I was in. And I was the only drug person pretending like I knew what I was doing. And somebody pulls me aside in the hallway and says, we really have a lot of interest to do colloidal chemistry in the oil field. And I was like, I have absolutely no interest in that. I got to publish my papers and get tenure. And they said, well, we have money. And I was like, oh, I'm in. So I think I learned my lesson in grad school, about partnering with industry and taking advantage of situations like that. So we ended up doing an 8-year project with ConocoPhillips on colloids that could deliver metals to the oil field as a well treatment, which that knowledge that I gained led me to understand how metals could be captured by Bond Biosciences to remove iron, which also led me to new materials for doing axioforce. With axioforce, we took iron filings, metal particles, and we had this idea that if you sprinkled them into rubber, you could make smart rubber. It's a pretty cool idea, right? You could take the Mr. Mustache metal filings and just sprinkle them into rubber. And then if you put something that recognized the movement of the magnet and the rubber, now you have smart rubber. And we got a project with Honeywell in Kansas City to make smart parts that could report if they've had an event, if they've been flexed or dropped or damaged in some way. And that was kind of the impetus of a new technology that eventually led to the founding of axioforce.
And so what are all the applications that axioforce is looking into? I know you have several on the market already. You have these force plates, but then you're also looking at measuring forces within apparel. So what are you most excited about?
So axioforce is the 8-year overnight success. We had a project with Honeywell for about 8 years, and Bryce was a big part of that project. And, essentially over time, we learned how much of the metal to sprinkle in, how magnetized it had to be, what are the properties of the rubber, and how do you pair that with what's called a Hall effect sensor. We actually all have them in the back of our phones. We have a magnetometer in our phones, and very cheap. And the movement of the rubber can be calibrated to a force. So I know it takes 3 newtons of force to displace it 1/4 of a millimeter this way, and 5 newtons of force to compress it 1/10 of a millimeter this way. And we're like, oh, that's awesome. You could put it in a helmet, and you could measure the impact in a helmet. We made little, what they're called load cells, but we put it into the 4 corners of a scale, and we found we could make a 3-dimensional scale, which those are out there. They use what's called a strain gauge that measures the flexing of a metal, and it tells you how much the resistance changes. But you can't drop a 3-dimensional scale. You can drop rubber. So we had this advantage in terms of durability, and ours were portable. And we still weren't quite getting the momentum in these different verticals we want. And then we found out that a 3-dimensional force plate costs about $10,000. And we could make ours for a lot less, like 100x less than that. So, suddenly we were cranking out 3-dimensional force plates, and we hired a guy who was at NASA to help with calibrating using a neural net, something I don't even understand, and an algorithm to calibrate these devices. And so now we sell force plates. We just sold a smart pitching mount where you put a force plate under the rubber and 2 force plates in the landing zone. We have smart batters boxes. We have smart golf setups. And we partner with companies that are already working in this space, and we sell them the intel inside. So we sell them the force plate solution to capture all this human kinetic data.
Yeah, that's really cool. So one of the things about your move to St. Louis that's been interesting is you haven't come by yourself. So you've moved a large family, but then you've got this extended family, and so we've got Bryce and Grant and Danny standing back there. You've brought
you guys are standing by each other.
You brought a whole team.
You're not in the lab anymore. You can spread out and mingle.
But maybe tell us about. I'd love to hear a little bit about your family moving here, but also about you've really been helpful to BioGenerator and to St. Louis in terms of you've brought all of this talent with you.
Yeah, it's interesting. So a lot of times the students and the staff, the postdocs, have ended up being like those that die on the hill for the company, the ones who are really willing to go all in on their technology and have interest in entrepreneurship. And that these 3 guys plus others really have been those people that they're willing to take a risk early on in their life. Maybe I can go work for corporate America later. but they want to see something happen, and they have a hunger and a passion and drive. And yeah, I mean, Danny came here about 3 years or so ago now, and it was a venture fellow position at BioGenerator that led him here, and then he got titrated into my businesses to help out. Grant, venture fellow at BioGenerator, kind of going through the same thing, and Bryce, working as a venture fellow at the OTM at WashU. These are the people that make the companies happen. I often joke that the guy who started the microencapsulation business with me, we were good at sort of, pretending we could do a good pitch deck, and telling a great story, and applying for a 9 Sigma award and getting $50,000 and high-fiving. But that's not operational. That's just screwing around. And so pretty soon they well, you have to file taxes, and you have your corporate documents don't look right. Then I'm like, what? What are you even talking about? And so it's the operational people and the technical people and the team that really make those types of things happen. So I'm just thankful that, some people in my circles have come over here too. We have a family of 7. We have 5 kids. So we have some budding entrepreneurs in that group as well.
Excellent. And thank you guys for everything that you're doing. And so we're working on a couple of companies right now, one with Danny and one with Grant, that are probably going to be part of this new venture studio that we're forming, HIFA. And we're working on this with Dave Smoller and Tom Cohen and Cory and I. And this is in collaboration with BioGenerator and Cultivation Capital. But the idea is to build research tools and services companies. And maybe if you can tell us a little bit about, why is that a good idea, and why is St. Louis a good place to do this?
Yeah, for me, the draw was it's not pharma. Because if you look at a financial projection on pharma or even med tech, it goes like this, and then it goes like this. And, maybe 10 years later or something, you're surfacing, and you're starting to have a chance to transact the business. I was trying to de-risk that by creating drug delivery companies that had technologies to partner with pharma. And so I think through all of that, I got some kind of indirect exposure to tools and services. You're partnering, you're working with other businesses, you're striking deals. And that is a way to generate income and revenue, so that you only go like this, and then you're starting to create, a positive spreadsheet. And so that was a passion for me, and it was fortunate to meet these gentlemen, Tom and Dave and yourself, at this period of time, because I feel like some of my companies have momentum, and I have maybe I don't have bandwidth, but I have passion for, HIFA and what we're doing. And then why in St. Louis? Sigma, Millipore, Thermo, a few pharma companies that are using these tools and services, a couple of behemoths that use a lot of tools and services. So I think it's really the right balance of being able to get to some kind of financial inflection point, and then be able to raise money smartly over time, grow the business, and be able to maybe even transact it locally.
So one of the things that's interesting about these companies is they tend to create a lot of jobs, and they tend to create them quickly because you actually have to deliver that tool or that service in fairly short order. So there's either manufacturing or service provision, that you have to build a team around, as we've done with Panome Bio and have done previously with Canopy Biosciences. And so, but it's interesting that, as opposed to building these on the coasts, that the cost of living, the cost of doing business, cost of operations lower here. One of the things I hadn't realized that Canopy's experiencing now that they're part of Bruker is retention, is that their retention is better here than at their other locations.
Yeah, I would echo all of that. You've heard the story, if you marry somebody from St. Louis, you're probably going to be back here at some point in time, and it matters where you go to high school, I guess. So this is all new to me. But yeah, retention and quality of life, it's a family town, all of those things, just provide a lot of gravity.
Yeah. So we're hoping to build, at least 5 of these research tools and services companies over the next 5 years, which will be exciting and fun to do it with you. So But Cory, let me ask folks to join me in thanking you for spending your time with us, and then we'll take a few questions. Great. So Christy is going to help us finding folks who have questions. I think there's one right next to you there by the column.
Hi. I just want to ask, like, is your company have any like production, like factory or site, and where is that?
Yeah, good question. So it depends on which company you're talking about. Good to see you again, by the way. The company that's making the drug, Bond Biosciences, it's manufactured in London, small suburb north of London. Kinemune is kind of outsourcing, so we're kind of pre-development. So we're still doing a lot of biology and taking advantage of resources that you can order in a catalog. And Axioforce, we like to use the word custom manufacturing. That's what you call it when you're not at large scale and you just got, the guys coming in at night putting together force plates and things. But our manufacturing's in Lawrence, Kansas. And they have to put in some longer hours sometimes to build force plates and calibrate them and then ship them out. I forgot to mention too, Axioforce is making smart insoles. So we have insoles that you can put in your shoes that tell you how much you weigh. Isn't that scary? And how much force you're putting into the ground when you jump, or if you're striding to hit a baseball or a golf ball or pitch, how much lateral force you're putting into the ground. And we got an NIH grant to put these on patients in a VA hospital in San Antonio. So we've collected data from about 5 patients so far. And it's exciting to think about smart insoles being used for neurodegeneration or gait assessment.
I think we have one here, and then Mike. Yeah.
Yeah, so with those, like the Hall effect sensors, are you measuring the force in real time as it's coming out? Or is it like you let it do its thing, then you come back later and try and inspect it?
It's a great question. You seem smart. Are you looking for a job? So right now we have a real-time Bluetooth communication with the smart insoles. Battery can be an issue. So battery only lasts about 5 hours of continuous collection. We've been working on kind of an algorithm that if you're sitting, they just shut down during that time, and then they start to wake up and recognize when you're moving again. If you're on Wi-Fi, it can last a day or a couple of days, and Bluetooth is high power demand. You can also do onboard storage of what you do through a session, and then that takes very little power.
With your friend who had the sarcoma that you had with Kinemune and you had the injection, where you put the injection, were you able to inject that into a sarcoma tumor?
Yeah.
Okay. And that showed inflammation, and did the tumor then recede?
It was only a 30-day period, and they only got a couple of doses before they had to get their standard of care tumor resection. Mm-hmm. So it wasn't enough time to see a statistical benefit in reduction of tumor size. So we focused on immune markers. We did see a downregulation of Ki-67. So there's some signals that there's things happening.
Hi. First of all, thank you very much for this interview. It's really a pleasure to listen to you. I'm surprised how many people still stand, so it means that, the skills of the presentations you master. I have more like general questions, especially since I see a lot of young people here, about your experience, and is there anything that you would do differently starting from your early stage over here when you are right now? Something that could simply make you progress faster or help to save some money, for example, or other things?
Thanks for that comment. First of all, I think BioGenerator has paid most of the people who are standing in the back, so thank you for amping up the attendance here. Anything I do differently, I almost always answer this question no, because I feel like even in failures, you learn. You should always be, the one thing you shouldn't do is not try. Unless I tell you not to try, and then you probably shouldn't try. It means like sometimes you should take no for an answer. But I feel like in my life, just the grind of doing the companies and trying has produced experiences, and then those experiences have led to this. And so I wish I knew more when I was younger, especially about how difficult it is to sell a business, to exit a business. But on the flip side of that, I might not have tried if I knew how difficult it was to grow and sell a business. So no regrets from me.
Eric.
Hey, Cory. We talked about this briefly a couple of years ago, but now that you're smart, insoles are more advanced.
I thought you were going to say now that you're smart. I was like, Eric.
No, I wouldn't do that to you. No, but now that you're a little more advanced, have you considered diabetic nephropathy, applications for diabetic nephropathy where people can't lose feeling in their feet because of nerve damage? I said nephropathy, I meant neuropathy, right? But where people lose sensation in their feet and don't realize that they're putting too much pressure on certain points and tremendous ulceration. So could those insoles be used to measure pressure points and see if there are sort of reproducible pressure points in the foot of a diabetic where the ulcers occur, which then might allow you to customize orthotics that address those specific areas in the feet?
My mind, I'm having a flashback moment. So I'm going to answer that question, but I'm going to amend my answer to the previous question. One thing I would do differently is I would be exposed to the financial side sooner, because I feel like I was treading water for a long time, and it's one of the reasons I kind of pushed my guys in the direction of getting exposure on the financial side. We tend to think primarily about tech and science, and that can be a dead end. We evaluated medical applications, and the feedback we got about diabetic neuropathy is that the reimbursement doesn't work. They only get about $100 to $150 a year to buy a new pair of shoes or two. I think you can buy 2 pairs of shoes per year, and it'll be paid for by Medicare. We can't make any money doing that. So then you'd have to do private pay, which is hard for the average person who has diabetic neuropathy. Plus, there's an approval path. You have to demonstrate to the agency that there is value in wearing these shoes as opposed to orthotic shoes. So it's dead for the moment. I do think over time, if we generate a data set that will help with prognosis, when are you at risk for fall? When are you showing signs of not having good sensation in your feet? When are you showing signs of like poor stroke recovery or potential risk of an event? Then maybe. It's just really hard to create those types of large data sets and compelling data and have the agency say, yes, we'll pay for that.
Let's do one last question and then we'll wrap up.
Hi. So similar to what he asked earlier, do you have any actionable steps for younger people who are interested in the kind of biotech space who are like science students?
Read, and then when you're bored, read. Most of my best ideas have come from outside of my field. So even as a graduate student, I made a point to read Scientific American, weird things that are completely unrelated to my field, advanced materials. I wasn't doing anything related with batteries, but I would get on the table of contents distribution list for a lot of the big impact journals. And I would, even just seeing the table of contents, you kind of develop a rhythm of what's important, what matters to people, where changes are happening. And then do that beyond science, do that with the investment community. So get on email lists for biotech investing, med tech investing, tools and services investing. And you'll find that you're just sort of like almost passively or indirectly absorbing information that gives you a wherewithal of stuff that's outside of like your myopic science project.
Great. Good advice. Great place to leave it. Thanks again.
Yeah, you bet. Thanks for the invitation. Yeah.